The topic of the lecture today is on development of fast, reliable, and automated isolation and fractionation methods for nanosized subpopulations of biomacromolecules. The structure of the lecture today is following. First, I will introduce the subject, why it's important. I will introduce the aims of this doctoral dissertation. I will introduce methods and techniques used in this thesis, and finalize with the results and conclusions that we have achieved during these studies. One of the important parts of why we're interested in biomacromolecules is that they take part in many different diseases and their formulation. So here is one example of lipoproteins, especially low density lipoproteins, which is accumulating as an atherosclerotic plaque over the years and might result in myocardial infarction. Another important particle for this dissertation, or particle type, is called extracellular vesicles, especially exosomes. And they play an important role in physiological functions such as heart functions, immune system functions, brain functions. But they also play an important role in pathological functions such as cancer, heart failure, liver diseases, Alzheimer's disease, immune system invasion. And they transfer different particles or metabolites and RNA inside them to other cells in the body. Here is one of the challenges that we have identified during our research. And for extracellular vesicle, this challenge has been that the sizes among extracellular vesicle subclasses, they overlap. So we see that there's different formation mechanisms for extracellular vesicles, but the particle sizes are the same. And another challenge is that in human body, there are also many different other biomacromolecules and nanoparticles of the similar size as extracellular vesicles or lipoproteins, and they also overlap in density, making density-based separation difficult, making size-based separation difficult. Now, conventionally, certain techniques have been utilized for the isolation of biomacromolecules, and these include size-exclusion chromatography. This is separating based on size. Ultrafiltration, which is also separating based on size. Immunocapture, which is separating based on function. Precipitation, which is also based on function. And ultracentrifugation, one of the most popular methods and techniques that is separating based on density. The limitation that we have found in literature and experience, for example, for ultracentrifugation, has been high cost. It's time consuming. It needs skillful operators. It requires tedious manual labor. It may result in batch-to-batch irreproducibilities. It might produce waste. It's prone to protein contamination, especially lipoprotein contamination in terms of extracellular vesicle. And also, since there's many steps where sample is prone to oxidation. Now, the aims of this research has been, first, to study antibody-antigen interaction, to develop affinity-based monolithic immunoaffinity chromatography methods to isolate extracellular vesicles based on this antibody-antigen interaction information, to utilize FFF coupled with multiple detectors to study and fractionate EVs based on the size, and to combine these two methods and techniques into an online automated system. Now, if we go further to the methods and techniques utilized in this dissertation, there were 4 main techniques. First was immunoaffinity chromatography. Second was asymmetric for fractionation. Then we utilized to study interactions, capillary electromigration techniques, and to study interactions, we utilized also quartz crystal microbalance. We also identified that there is a need for methods that address function and size of biomacromolecules at the same time to result in these pure subpopulations that we are interested in, especially for future clinical, diagnostic, therapeutic applications of biomacromolecules. Specific goals for development of isolation methods in our case has been speed of the method, reliability of the method, selectivity, reproducibility, automation, ability to separate subpopulation, and also suitability for complex biofluids such as human plasma. We utilize different antibodies to capture lipoproteins and extracellular vesicles. And you can see in this figure how they place in different papers in this dissertation. Now, the first technique was called immunoaffinity chromatography. And the basic principle of this technique is that we introduce biological sample inside a monolithic column that has immobilized antibodies inside the column pores. And the principle is following, that the biological sample travels through the pores and the antibodies capture the biomacromolecules of interest. And then biomacromolecules can be eluted from the pores and collected for further analysis. With this technique, we reach certain specific goals that we had for isolation methods, which were speed, reliability, reproducibility, selectivity, and suitability for complex biofluids. Now, to study interactions between antibodies and antigens, such as lipoproteins, we utilize capillary electrophoresis. And one of the sub-techniques of capillary electrophoresis called partial filling affinity capillary electrophoresis. In this technique, we hydrodynamically introduce the samples, first the analyte into the capillary, followed by receptor. And by applying voltage, these two particles migrate and form a complex. And with this complex migration, we can then deduce the affinity information. Another technique which was important for us in the study of antigen-antibody interaction has been quartz crystal microbalance. And in this technique, we have immobilized on top of the quartz crystal microbalance chip antibodies and then associated lipoproteins to these antibodies. And the association results in a frequency change, which is registered. And with this frequency change information, we can then calculate also interaction information. And in this thesis, we utilized a tool called rate constant distribution calculations or interaction maps. But we have also developed better tools later. Another technique which was important in the size-based separation of biomacromolecules has been asymmetric flow, field flow fractionation. And this is a technique where biomacromolecules are separated by two different flows, one flow being cross flow and the second flow being normal parabolic flow. And by manipulating these two flows, we can focus particles and they separate based on their diffusion, bigger, sedimenting faster, closer to the membrane, and smaller, slower. And when we stop the cross flow, the particles start traveling, and in the normal mode, smaller particles reach the detector first, followed by the bigger particles. And we get this nice size-based separation. And with this technique, we reached goals which were speed, reliability, reproducibility, automation, and ability to separate subpopulation. Now, the main results, or the main result of our studies and this dissertation, is the development of automated online immunoaffinity chromatography asymmetrical field flow fractionation system. And in this, we combine the affinity and the size-based separation together in an automated fashion. And with this, we reach all of the specific goals that we have set for ourselves, and also additional goal, which was to prevent oxidation, because the system is not in touch with oxygen at any part. The system consists of pump, autosampler, that's selection valve that control the entrance of the sample into immunoaffinity chromatography column. Then it has 6-port valve that is introducing the isolated sample into the FFF channel. And then in the end, we separate by size and detect by different detectors, which already give us important information on concentration and sizes. And in the end, we collect the fractions for further biological assays to be done. Here's a simplified illustration of how this instrument works. So first, the biological sample is introduced into a monolithic column, and it captures the biomacromolecule. Then we wash the unwanted protein from the column. And in the end, we elute the biomacromolecule from the column inside the FFF channel, which in turn enters automatically the sample into the FFF injection and size fractionation. Then at the same time, when the FFF starts doing the fractionation based on size, we can prep the monolithic column for next round of isolation, and thus run in sequence the isolations and fractionations. And here we see the separation based by size. And first, the smallest particle arriving due to them being in normal mode, or FFF being in normal mode, and the bigger ones follow. Now here is example of the results that we obtained while testing our system. Since we had quite good experience with lipoproteins, we utilized LDL as our model sample in doing the optimization of the system. As you can see, we reached quite impressive results, especially in terms of reproducibility of the isolations, in terms of size and in terms of concentration. Then, very important, we applied this technique also for the EV isolation and fractionation. And we reached very interesting results applying 2 different antibodies. We showed that we could isolate with many different antibodies extracellular vesicles, and extracellular vesicles in the size of exomeres and exosomes especially. And exomeres is one of the new particles that has been found utilizing FFF in 2018, which is yet to be studied for its further physiological meanings. Another important detector that we introduced was diode detector that is especially helpful in the study of complex biological biofluids, because, as I said before, there is a problem of lipoprotein contamination in conventional isolation techniques. So we needed to be sure that this doesn't happen in our technique. So we did run plasma samples and isolate them, especially for lipoproteins. And as you can see, when you isolate for lipoproteins, lipoproteins contain a peak, which is from wavelength 420 to 520. And this is representing the carotenoids present in the lipid core of the lipoprotein particles. And if you look at the picture below, you don't see this carotenoid peak. By this, we could conclude that that lipoprotein contaminants are not either present or in very little concentration in our isolates. Now, to conclude this lecture, I would say that we have introduced a couple of new methods and techniques, and especially developed this automated online immunoaffinity chromatography asymmetric field for fractionation system for the isolation of subpopulations of biomacromolecules. We can process 18 to 38 samples in 24 hours with minor operator involvement. And for the first time, we could isolate EVs in the size of exomeres for human plasma. This has implications in future potential in areas of clinical research and diagnostics, and maybe even therapeutics. In addition, we found QCM and partially filling affinity capillary electrophoresis very helpful in selection antibodies and studying antibody interactions.